Now, what's super important to understand about cellular respiration and your aerobic versus anaerobic respiration is we do have to think about the organelle that everyone fondly knows as the powerhouse of the cell, and that is the mitochondria. So, if we look on the left-hand side of this model, what we have is we've got the cytosol. And then if we look on the right-hand side of this model, well, we have kind of and whenever I'm talking about the model, I'm talking about the mitochondrial membranes that are depicted here. So what we've got is let me get this, we've got the cytosol. Okay. So that's kind of your within the cell. And then we've got basically this space right here. Now, this space right here where I'm putting my second and third boxes, what I'm really indicating, and this is kind of the interior of the membrane. Now, what makes the membrane a little bit complicated is that it has two important membranes. It has an outer membrane and an inner mitochondrial membrane. Now, within or between them, I always kind of think about this similar to how I think about like if you imagine like a bicycle wheel. Okay. A bicycle wheel has a tube on the outside, and then it has what's known as an inner tube on the inside of it. Where you pump all the air goes into the inner tube. Now, what that means is that as you inflate the inner tube, that will expand and that will fill the space within the tube. Okay, so if we think about our mitochondria as the mitochondria as the bicycle wheel, we've got the tube, which is our outer membrane. Now, that outer membrane, what's important about it is that it has openings. It is freely permeable up to 10K. Now, what that 10K means is 10 kilodaltons or 10,000 daltons. The inner membrane, however, is only permeable to water, CO2, O2. That's it. So it's very, very selective. That inner membrane, in order for something to pass through it, it has to have a transporter. It has to have a mechanism to bring it directly across. So what that means is it's selective. If that membrane is not present, or if that selective protein is not present, well, then that thing's not getting from one side to the other side of the membrane. And what that means is that, well, our mitochondria will keep a lot of stuff out of it. Now, this illustrates the size of and kind of the relative selectivity of our two membranes. The outer membrane is not very selective, the inner membrane is very selective. So one of the things that's a little bit complicated is I was just showing you guys the um, citric acid cycle, the tricarboxylic acid cycle, the Krebs cycle. Well, pyruvate is involved in that, and pyruvate is a molecule that is transported into the mitochondria because every step of the citric acid cycle takes place within the mitochondria. Now, here is a kind of crash course of all of that. So I'm going to go through this and spend a little bit of time on this because this is a very important and kind of comprehensive cycle. So we have just done this right here. We have produced two pyruvate molecules, which are 3 carbons each, from one 6 carbon glucose molecule. In addition to that, we produced a net of 2 ATP and 2 NADH. Now, those NADH are going to be transported into the mitochondria using a protein complex known as the malate aspartate. So what you need to know is that the malate aspartate shuttle is one of the protein complexes that, let's see, well, it translates, let me say translates as opposed to moves. It translates the energy of NADH across the membrane. Another protein that does the same thing is known as the glycerol phosphate shuttle. These are protein complexes that you'll learn more about in metabolism. Okay, so where this NADH total, when you look at here, it says 8 NADH. How did we go from 2 to 8? Well, what's not being accounted for there, or sorry, what is being accounted for there is the um, citric acid cycle and the NADH that produces. So, in summary, what you guys need to be familiar with is what do we get essentially from one glucose molecule? Well, one glucose molecule gives you two NADH and two ATP from glycolysis. After it's all said and done, glycolysis and the citric acid cycle, you get six CO2. So remember, six carbons in C6H12O6 glucose. We get six CO2 from that. So that is the complete oxidation of glucose down to six CO2. We also get eight NADH, two FADH2. Okay, so I've kind of reframed this because one of the things that I'm not really, not to say I'm not accounting for it, but it's just not as high of a priority is the ATP. And this is one of the reasons that I think that metabolism is really fascinating because, if you go to, say a high school science class and you ask, what's the energy currency of your body? They'd say ATP. And they're absolutely right. Um, but when it comes to complete oxidation of glucose down to CO2, well, yeah, ATP is involved. You get ATP from the anaerobic metabolism or the anaerobic breakdown of glucose. But what really matters is NADH2 and FADH2 because these two compounds are energy or electron carrying molecules and they will be used in something known as the electron transport chain, which is what we've got going on right here. Okay, so this shows basically our breakdown of glucose to CO2 and what comes from that. Now, do I want you to spend a lot of time on the citric acid cycle? Not particularly. Are we going to spend a lot of time on it? No, I've pretty much gone over the important points of it all. Um, from 2-pyruvate, you're going to get 6 NADH and 2 FADH2. So that's what you're going to get all together the um, from the complete oxidation of glucose. Now, the rest of this is important and it is relevant because you're going to generate a lot of ATP, but you've got to get it, or it's going to be generated in the mitochondria, but you've got to get it out of there. So that's what we've got down here. We do have something that moves ATP out of the mitochondria because, well, if we just make a bunch of ATP and it stays within the mitochondria, it's kind of useless to us. But that's something, for another day, and that's something for another class. So what we're going to focus on here is we're going to focus on kind of what we referenced and alluded to previously in the form of, well, yeah, you can get energy from ATP or from glucose, but you can get energy from lipids. Now, when we talked about lipids previously, we talked about the five different classes: sphingolipids, glycerophospholipids, triacylglycerols, fatty acids, and cholesterols or steroids. Um, there were your polar and your non-polar lipids. Your non-polar lipids being your fatty acids and your triacylglycerols, those are the energy containing lipids. Now, they are a major part of membranes, or they are a contributing part of membranes, but they're an energy store. So from 1 gram of a lipid or of just kind of a general lipid, you get 36 kilojoules of energy. Now, that is approximately 2 times that what you get from the same number of grams of a carbohydrate, which if you think about it, well, one aspect of that is the fact that we've got um, all of our molecular mass of a lipid versus all of our molecular mass of a carbohydrate. We're going to get more of your molecular mass of a lipid is wrapped up in carbons and hydrogens compared to the molecular mass of a sugar, which has carbons, hydrogens, and oxygen. So lipids do provide a huge amount of energy, and they're very valuable for that purpose. 30 to 60%. This is typical guidelines. Of course, this is not gospel or, I guess, a endorsement. because I think that this can vary quite widely. But most people, the caloric intake is a range of about 30 to 50% of your calories come from fat or come from lipids. And those lipids are going to be either triacylglycerols or triglycerides or fatty acids, which indirectly which triacylglycerols or triglycerides are, well, glycerol plus a fatty acid. Now, 90% of dietary lipids is in the form of triacylglycerols. So that's glycerol plus fatty acids. And if you remember, we have the omega fatty acid numbering scheme and we have the delta numbering scheme. And that's depicted here at the bottom. So just a little reminder of what a triacylglycerol looks like, also known as a triglyceride. Now, that's what we've got at kind of a molecular level, but how do we get there? How does your body get there, essentially? Because if you look at, these things shown below, they're little chocolates, they've got lots of sugar. They've also got a good amount of fat. And that fat is in the form of triacylglycerols. So how do they get there? So this is the rundown of everything. We're going to break this down into individual pieces so we can process it a little bit better. Much like how your body takes dietary fat and breaks it down into individual pieces. So when fat gets to your stomach, it triggers the production of compounds known as bile acids. Bile acids are produced in the liver and they're stored in the gallbladder. Now, we see dietary fat go into your stomach, which inspires the production of these bile acids. The bile acids are represented by these black dots. Those go from the liver to your gallbladder, and then they enter your stomach. Now, it's tempting to think about a bile acid as, or for me personally, whenever I hear the word bile acid or bile salt, for some reason my mind immediately goes to sodium chloride. Because whenever I think of a salt, I always think of table salt. Now, a bile acid, that's kind of a misnomer, maybe not a misnomer, but try to make yourself not go in that direction, but instead think about these as compounds that are made up of cholesterols. So here's our fatty acids and what they're broken down into. And here's our classification again. Cholesterol is where I wanted to go, and that's what I really want you to think about, because a cholesterol compound, well, that is the precursor, basically, for the synthesis of a bile acid. So bile itself acts as a detergent. They are cholesterol derivatives synthesized in the liver and stored in the gallbladder. Now, examples of bile acids I want to show you are shown at the bottom of the screen. And so notice what we have here is our cholesterol molecule in the middle, in that basic structure of a cholesterol molecule, the four rings, the 3 6-membered rings and the 1 5-membered ring. Now, bile acids or bile salts, well, they are that, but they have now what makes bile acids and bile salts different from one another is these different positions, R1 and R2. Depending on what is there, if R1 has an OH or an H, those are basically the two different options as what can be there. Now, R2 by comparison can have an OH. I can have this group right here, have this group right here, in which case we've got a matrix here essentially. If at position R1 you have an H, and then at R2 you have OH, well then that's chenodeoxycholic acid. If at the same time you have an H in the R1 position and an R2 position, you have that NHCH2CH2 SOH3 or SO3H2, you have taurochenodeoxycholic acid. So there's a number of different bile salts and bile acids that contribute to the breakdown of fatty acids. But one of the things that they do is they act as a detergent to help break up larger particles to make smaller particles known as micelles. These are spherical. They have lots of fatty acids within them. And I want you to notice whenever you look at those molecules, you have a polar head group with a singular tail. So that polar head group is that carboxyl group, and then the tail is just your um, alkyl functional group. Now, these bile acids are going to interfere with fatty acids. They're going to help that dietary fat break down. Now, those large lipid droplets will interact with your bile acids, which is kind of what we're seeing begin to happen here. Maybe I can pick a different color there. It's what happens or begins to happen here, continues happening, and then, well, it's continuing to happen down here in your small intestine. So those large lipid droplets are broken down with the help of bile salts into smaller lipid droplets in smaller micelles. Now, those micelles are going to contribute to that, those bile salts are going to contribute to the formation of micelles, which those micelles are going to also contain some triacylglycerols, which those triacylglycerols will be broken down into individual fatty acids through the process known as hydrolysis, conducted by a lipase. Now, what I think is valuable to recognize here is that any triacylglycerols that exist in your small intestines will eventually see this same fate. They'll be broken down into individual fatty acids, which we see right here. And then they will be, this sounds kind of silly, but they will be repackaged into triacylglycerols in the intestinal epithelial cells. Once that happens, they will, I gotta clear my drawings. They will go on to basically make their way into the circulatory system. But a fair question, when you think about a fatty acid, is to recognize that this is a molecule that, best case scenario for myself. But overall, triacylglycerols and lipids in general are going to be fairly insoluble because of their nonpolarity. Now, so those triacylglycerols will be reformed into, or broken down to fatty acids, then reformed as triacylglycerols. But how? Because we've got something that arguably doesn't like water. Now, how do enzymes deal with these big chunks of lipids? They're insoluble in water, and like I said, they form micelles at best. Now, enzymes, by comparison, are going to be soluble in water. Enzymes like where they have all their nonpolar insoluble stuff in the middle, but then their periphery, their surface exposed amino acids are polar. Now, in order for that to happen, in order for a individual enzyme to take a triacylglycerol or interact with a triacylglycerol or a fatty acid for that matter, these intestinal lipases, what they will do is they have a structure known as a lid that opens to expose their hydrophobic active site, because the enzymes of the active site have to largely be hydrophobic. And so that's how they'll break them down. But the important part about the interaction between an enzyme and a lipid is what's known as the interfacial activation. The lipid-water interface. Lipases exhibit interfacial activation, meaning their enzymatic activity increases when they come in contact with the lipid-water interface. So lipases act at the surface of these lipid droplets. So they have this interfacial activation, this water-lipid interface, where basically they're able to interact and open up and pull one of these either triacylglycerol or fatty acids out. Well, triacylglycerols pull those out so that they can break them down into individual fatty acids and glycerol. Now, that's what's happening down here in order to make these compounds known as chylomicrons, which can circulate through the body, which have cholesterol, which can then be basically transported to different locations. Now, this is not a schematic that I want you to be super concerned with and aware of. I do want you to understand the role that bile acids play and kind of what they're structured from. But the details of where different things go, I'm not terribly, that's not something that I want you to spend a lot of time on. Now, if you look at this structure right here, this schematic, this is showing a specific lipase known as a hormone-sensitive lipase. And this is a way that signaling, biological signaling can take place. So if you look in the upper left-hand corner, we have a hormone. And that hormone could be epinephrine or it could be glucagon. Those are both hormones that are involved in fatty acid metabolism. Now, what those specific hormones are is not a high priority. You'll learn a lot about hormones and their interplay in metabolism in metabolism. But you could think about hormones like epinephrine and glucagon as being involved in, well, let's turn on breakdown of this or let's turn off breakdown of this. So a hormone is a molecule that will be perceived by a protein known as a receptor protein. Now, here what we have is a receptor protein that perceives a hormone. So this receptor protein perceives the hormone. So that's a membrane protein. Think about this receptor as being associated with the lipid bilayer of a cell. Now, this particular cell type is an adipocyte or an adipocyte. It's a fat cell. What this receptor protein is going to do is it's going to activate a protein complex, or G5 signaling protein that's going to activate something known as adenylyl cyclase. Adenylyl cyclase will convert ATP into cyclic AMP. And so in the process, it's going to pluck off a pair of phosphates. So this cyclic AMP is then going to go down to turn on a protein complex or a protein known as protein kinase A. Okay, so essentially what we've got is a hormone that is perceived. This hormone is outside of the cell. This membrane protein, this receptor membrane protein, it perceives that hormone, then it leads to the activation and production of cyclic AMP. Cyclic AMP activates protein kinase A, and protein kinase A is going to, what it does, it is a transferase. It's going to use ATP to phosphorylate hormone sensitive lipase. And you might be thinking, okay, I've heard about transferase. Yes, you have. We talked about transferases in the context of kinases. Phosphofructokinase 1 is a transferase. Okay. Now, what's interesting about transferases is they can either be targeting, or one of the things that they can do is they can be targeting a protein or they can target a sugar. Phosphofructokinase takes ATP and adds it to fructose 1 or fructose 6 phosphate. So it transfers that phosphate onto there. Alternatively, protein kinase A is an enzyme. It is a transferase. It transfers a phosphate group onto the enzyme hormone sensitive lipase. So protein kinase A is a protein kinase because it is targeting another protein. Where is that phosphate ending up? Well, that phosphate that protein kinase A is transferring to hormone sensitive lipase is ending up on something like a serine or a histidine, for example, or a threonine or a tyrosine. Those are all great targets for a phosphate group to end up on. So when hormone sensitive lipase is phosphorylated, it is activated. And what it is going to do is it is going to relocalize to a lipid droplet. And what it's going to do is it's going to go to that lipid droplet and it's going to pick up a triacylglycerol and it's going to break it into individual fatty acids. Okay, great. Now there's another thing that protein kinase A is doing. It is phosphorylating this protein that basically is wrapped around a bunch of lipids or wrapped around on the exterior of a lipid droplet. That protein is known as perilipin. When perilipin is phosphorylated, which is what we see right here, when perilipin is phosphorylated, then the landing place for hormone sensitive lipase is more available. So protein kinase A plays two roles in the metabolism of fatty acids. It causes perilipin to be reorganized, it also causes hormone sensitive lipase to go to the right location. So PKA does those two things. PKA activates perilipin, it also activates hormone sensitive lipase. Hormones are going to do that, but upstream of it. So this hormone that I'm putting another, and one more box around that hormone up there is doing all of this, not just indirectly, but it's like indirectly it is activating something indirectly, that is activating something indirectly, that is activating something directly. Now, there's a lot involved here, but the entire purpose of that is to, within an adipocyte, break up fatty acids into individual molecules that can then be transported within the bloodstream. So when they are free in the bloodstream, they can be transported to a myocyte where they can be transported into that cell where we can undergo a process known as beta oxidation, and we can generate a bunch of ATP from that. Now, a lipid or a fatty acid is not going to be quite so freely available. It has to use serum albumin as a transport mechanism. So it has to find a serum albumin molecule protein to migrate throughout the bloodstream. Now, that's a lot going on there, but that's kind of the summary of what's going on there. Okay, now this is one of the things that I alluded to previously, the glycerol, or I alluded to the glycerol 3-phosphate shuttle. But for this, what I want you to know is that glycerol from a triacylglycerol is going to end up in the liver. Now, in the liver, glycerol is converted to glycerol 3-phosphate or 3-phosphoglycerol. We can convert that with the help of FAD to dihydroxyacetone phosphate and FADH2. That DHAP can be then converted to glucose. Now, the liver is what performs the conversion from pyruvate and all the steps in between back to glucose. That process is known as gluconeogenesis. So if if you know someone who eats a, high fat diet, they still do have circulating glucose within their bloodstream. They've got glucose for their brain so that they can continue, moving around. Then we have to use something known as acyl-CoA or coenzyme A, and we have to add a acyl group to it. Before we can actually burn a fatty acid, they must be activated by being attached to coenzyme A or CoA. Now, if we look in the lower right-hand corner of this slide, what we have is our acetyl-CoA molecule. Now, I'm going to take a couple of different squares in order to draw this, but this and this make up coenzyme A. What that means is that the alkyl chain of a fatty acid, that's that right there. So we have to take a fatty acid, add coenzyme A and ATP to make FA-CoA, which I'm going to go ahead and change colors to highlight FA-CoA. This whole thing is FA-CoA. So in order to make this, we use a ligase. Remember the other day we had kind of a newly revised and enhanced definition of a ligase, where we basically used ATP and harnessed the energy of that in order to make something. So here what we have is effectively a ligase, where we break ATP into AMP and PPI. So we break that high energy bond in order to make something. So this FA-CoA, we used ATP in order to do that. Now, let me clear all this out. So there's certainly a lot involved in the metabolism of fatty acids and kind of getting it all ready to go. We've talked about breaking those triacylglycerols down. Now we're going to get into, all right, we've got a free fatty acid, or we've got an FA-CoA. Let's take care of that. Now, lipid oxidation occurs in the mitochondria, and lipid synthesis occurs in the cytoplasm because there are times that we need to store energy as a fat. But then there are other times we need to take a fat and break it down. So if we're going to be trying to get energy from a lipid, we're doing that in the mitochondria. If we're trying to make a lipid for the purposes of, a membrane or just storing the molecule, that's going to take place in the cytoplasm or the cytosol. Now, if anyone ever drinks like a Monster energy drink, if you look around the rim of the bottle, it usually says like taurine and sometimes sometimes there's creatine in them. But one of the things that you've probably also seen on there, and if you haven't yet, look around, you'll see this compound right here, carnitine. Carnitine looks like creatine, but it is not the same thing. So carnitine is a compound that carries a fatty acid into the mitochondria so it can actually be broken down. This process right here shows an example of a carnitine acyltransferase. This one is specifically carnitine palmitoyl transferase. Carnitine palmitoyl transferase 1. What this is going to do is it's going to take a FA-CoA, specifically of palmitic acid, which I think is 12 carbons long, and we're going to transfer this FA onto carnitine, and we're going to make a carnitine CoA. Carnitine palmitoyl transferase does that. So it removes the FA and slaps it onto the carnitine. The resulting FA carnitine molecule will be trafficked into the mitochondrial matrix. So within that very selective membrane, using a carnitine carrier protein. This carnitine CoA will then be acted on by carnitine palmitoyl transferase to reproduce free carnitine and our FA-CoA. So essentially what we have is carnitine is a molecule that the carnitine is a compound that whenever it has an alkyl chain, the carnitine carrier protein says, I recognize you. I'm going to transport you into the matrix, into the mitochondrial matrix. Free carnitine, the carrier protein says, I don't know you. Whenever you have FA carnitine, boom, it gets transported in. Free carnitine does not. Free carnitine gets transported out. So this is carrying the alkyl chain that we are ultimately going to break down for ATP. Now, carnitine acyltransferase is inhibited by a compound known as malonyl-CoA, and malonyl-CoA, or the reason for that, is that malonyl-CoA is used for fatty acid synthesis. So if malonyl-CoA, which is used in fatty acid synthesis, is present, well, that's going to block this transport, which makes sense because you don't want to be transporting something to be broken down if you're also trying to make that something. Now, this is a nice schematic of beta oxidation. So this shows our fatty acyl-CoA up at the top. Now notice this molecule has a number of carbons on its alkyl chain. And I'll go here, this is my alkyl chain. Notice how we've got our alpha and our beta carbons highlighted. Okay. The reason that we call this beta oxidation is because we are really kind of focused in on this beta carbon. We want to put a double bond between our alpha and our beta carbons. And we want to break down and remove 2 carbons at a time from a fatty acid chain. This beta carbon, when we're all through with this, is going to be this carbonyl carbon. So what I mean by that is we're going through rounds of this. And in the first round, we're going to pluck off these 2 carbons. And those 2 carbons are going to form this molecule along with acyl-CoA or coenzyme A. It's going to make acetyl-CoA. Acetyl-CoA is a compound that's methyl group plus a carbonyl carbon plus coenzyme A. So our goal and our objective is to make that acetyl-CoA. Now, that's the way that I always think about this kind of math and this accounting is our fatty acyl-CoA has a certain length, and we're going to pluck off 2 carbons at a time. There's basically 4 steps. The 4 steps are acyl-CoA dehydrogenase. And that first step So our first step is catalyzed by this acyl-CoA dehydrogenase. What we are doing is we are adding a double bond between our alpha and our beta carbons. Okay. Then what we need to do, or sorry, in order to do that, we're generating this compound right here, known as FADH2. This is taking place in the mitochondria. That FADH2 is something that will feed into the mitochondrial electron transport chain. Now, this part of the whole mechanism and this whole pathway is a little bit, I'm always a little bit complicated as to whether or not to include it because we will later get to the electron transport chain. I'll introduce it. And effectively, all of that is a part of the electron transport chain. So if you wanted to think about beta oxidation in a step or the steps of beta oxidation or one round of beta oxidation, I think the simplest way to approach it is to say, okay, well, in step one, I generate FADH2. And everything that I've got that's blocked out with this really dramatic pink, just ignore it for the time being. So we've removed, and if you think about FADH2 and if we think about our fatty acyl CoA as well as trans delta 2 enol CoA, our resulting molecule, what we did was we removed some electrons from it. So we have FADH2 as a compound that is now carrying a bunch of electrons. So we removed electrons and in the process, place this double bond right there. Okay. Now, after we've removed those electrons, what we need to do is add water. And the enzyme that's going to do that is enol CoA hydratase. It's going to add water across that double bond, and we're going to end up with this rascal right here. So I always like to block out a portion of this structure, and I'm going to do that right now, because what we know that we're trying to make is down here in the lower, it's kind of in almost dead center. I've got it underlined with a big black box. There we go. If you look at what is highlighted right here, what is underlined right here, we are beginning the steps of getting that acetyl-CoA. And that acetyl-CoA is kind of what we want to make in all of this. So, get rid of that, get rid of that. So when we add this water across this double bond, we make the 3L-hydroxyacyl-CoA. So if you want to think about the steps of beta oxidation, remove electrons, add water. Okay, those are your first two steps. The third step is going to be, well, let's look at our enzyme name, 3L-hydroxyacyl-CoA dehydrogenase. Now, if you think about the classification of enzymes and you think about dehydrogenases, well, dehydrogenases are the enzymes that are involved in, well, oxidation reduction reactions. So a dehydrogenase is going to be a compound that, or an enzyme that makes or utilizes or makes, well, it's involved in oxidation reduction reactions. We're removing electrons from this 3L-hydroxyacyl-CoA, and in the process, we're making this beta-ketoacyl-CoA, which I'm going to put a box around. And we are also making NADH. Okay, so that NADH, that's our electron carrier. So we've gotten, let's see, two electron carriers, FADH2 and NADH now, and we've added water. So we've put a double bond in, or we've removed electrons, added water, removed more electrons. And if you're thinking, okay, removing electrons, sure, that's fun. Remember to think or try to think back to if you have a potassium atom and you add it to water. That potassium atom has electrons and it will release them in a very dramatic fashion. And there will be kind of an explosive dramatic fashion. Okay, so our very last step is to take that beta-ketoacyl-CoA and cleave off acetyl-CoA, leaving behind our fatty acyl-CoA that's 2 carbons shorter. Okay, so these 2 carbons, which I already kind of highlighted as, hey, we're kind of digging the groundwork to remove 2 carbons. I've got them all highlighted. So this part of our molecule is going to be cleaved off. But only now do we have a carbonyl group that can basically be similar to this carbonyl group. We'll use the enzyme beta-ketoacyl-CoA thiolase as well as coenzyme A. Coenzyme A will add on to that carbonyl carbon, will release acetyl-CoA, and now we'll have that fatty acyl-CoA that's 2 carbons shorter. We'll continue doing this until our 16 carbon molecule is down to nothing but 8 fatty acyl-CoAs. Now, an important thing to think about with this is the math. Okay, so we went from, this doesn't say the specific number, but think about a round of beta oxidation taking 2 carbons off at a time. So what that means is that if you have a 4 carbon long molecule, one round will take 2 carbons off. If you're left with 2 carbons, well, those 2 carbons are not just 2 carbons floating around, but instead they are a fatty acyl-CoA. So from a, let me type this up, a 4 carbon fatty acid will be split into 2 acetyl-CoA molecules after 1 round beta oxidation. So if that's what a 4 carbon molecule does, what about an 8? Okay, so 8 carbons, 1 round, after 8 carbons after 1 round is 1 acetyl-CoA and a 6 carbon fatty acid or fatty acyl-CoA after 2 rounds. Now we've got 2 acetyl-CoA and a 4 carbon fatty acyl-CoA. After 3 rounds, this is where it's kind of complicated. After 3 rounds, you have 3 acetyl-CoA but wait a minute. You actually don't have 3 acetyl-CoAs. You have 4 acetyl-CoAs. Because when you take a 4 carbon long molecule, let's use my fist to represent 2 carbons. If you split it in half, well, here's our acetyl-CoA, which is, I'm going to use the highlight, that. But then our last enzyme, beta-ketoacyl-CoA thiolase, adds coenzyme A. So in effect, we have another acetyl-CoA. So that's one of the complicated parts about, okay, how many rounds does it take to do this? This is the exact same thing, just giving another specific or another more detailed visual. So we remove carbons, add water, remove electrons, add water, remove electrons, add coenzyme A. That's it. Remove electrons, add water, remove electrons, coenzyme A. And you rinse and repeat over and over again. Now, your mitochondria have 4 different acyl-CoA dehydrogenases used for fatty acids of different lengths, ranging from 4 to 18 carbons long. Ranging from 4 to 18 carbons long. Deficiency of medium chain acyl-CoA dehydrogenase is associated with SIDS, sudden infant death syndrome, about 10% of the cases. Now, beta oxidation, all of this takes place in the mitochondria. To visualize kind of the breakdown of a fully saturated fatty acid, which is kind of the range in which we're going to focus for this class, because they can get a little bit more complicated. We do 7 rounds. Okay, that might sound like that doesn't make any sense because it's 2 carbons at a time. Well, remember that very last round. This 7 rounds of beta oxidation. So from 7 rounds of beta oxidation, you're going to get 7 FADH2s. Those FADH2s are going to be converted to 14 ATP. 7 NADH will give you approximately 21 ATP from your 8 acetyl-CoA molecules. That's going to give you, because those acetyl-CoAs, that's 2 carbons plus the coenzyme A, that's going to go through the citric acid cycle. It's going to give you 8 GTP, which is the equivalent of 8 ATP. 24 NADH are going to give you about 72 ATP. 8 FADH2 are going to give you 16 ATP. So from a 16 carbon long fatty acid, you're going to get about 131 ATP. That's a huge, that's a huge amount of ATP to get. If you think about a glucose molecule is 6 carbons. Okay, from those 6 carbons, you're going to be able to get, let's see, 2 acetyl-CoAs. So you'll get 2 GTP. Long story short, the amount of energy, the amount of ATP that you get from fats is much greater than that of sugars and glucose. You do, however, have to pay back in order to activate a fatty acid to a fatty acyl-CoA. You've invested ATP to do that. So 129 ATP will come from 7 rounds of beta oxidation or from a 16 carbon long fatty acid. Is the amount of energy that you get from an unsaturated fatty acid going to be different? Absolutely. There's no doubt about it. Now, just as a little bit of a review, we've broken glucose down to CO2. We've generated some NADH in the cytosol through glycolysis. We need to use that NADH to produce more ATP, but how? In order to do that, we're going to go through something known as the electron transport chain, commonly known as the mitochondrial electron transport chain. Now, this is very similar to the process known as photosynthesis, because in photosynthesis, you're moving electrons, and you're moving those electrons around, and you're getting a lot of the production of ATP from it. Now, essentially, what you need to be aware of is what's in the matrix and what's outside of the mitochondria. So we have NADH that we've generated through beta oxidation. So we have this NADH that's accumulating in the mitochondria, and we're going to send that through these different complexes. Now, these complexes are known as 1, 3, and 4. There's another complex number 2, and that is going to be the way in which FADH2 generates ATP. Now, the takeaway of all of this is, let me go ahead and highlight a couple of things. This is a protein complex. This is a protein complex. This is a protein complex. This right here is a electron carrying compound. Throughout all of these complexes, what is taking place is a series of oxidation reduction reactions. So electrons are being picked up by one thing, so they are being reduced, and then they're being oxidized by transferring those electrons to something else. It's almost like if you've ever played a game of telephone or something like that, or made a long line of people and held hands, and whenever somebody squeezes someone else's hand, or whenever your hand gets squeezed, then you squeeze the opposite hand. You squeeze the hand of the person next to you, and you send that signal through. That's effectively what's happening here. All the people in that long line are all of the different electron accepting and donating species within these complexes. So NADH is the first electron carrier, dumps electrons off at complex 1, and then circulates through. The important, or maybe the most important part of this all, is that this transport of electrons is going to lead to the movement of protons to the intermembrane space. NADH2 is going to move a total of 10 protons across this membrane. Now, FADH2 plugs in basically right here, where I have that red square that doesn't have anything in it. FADH2 is going to lead to the production of 6 protons being pumped into the intermembrane space. So the difference between FADH2 and NADH is NADH starts at complex 1, FADH2 skips complex 1. So these are our different complexes that are doing all of this work. Now, NADH moves 10 protons, FADH2 moves 6 protons. Okay. It's difficult to think about this in terms of, oh, one proton causes a change in pH, but that's exactly what it is. When protons end up in the intermembrane space, that means the pH within the intermembrane space has gone down. Why has the pH gone down? Well, because the hydrogen ion concentration has gone up, the proton concentration has gone up. So the pH is lower, it's more acidic there. Now, what that's going to lead to is this protein right here, known as ATP synthase. If you look at the bottom of this figure, that is showing the movement of protons within this protein complex, where a proton could conceivably make its way across the membrane, but instead what it does is it drives this ATP synthase, this protein, which the bottom portion of it right here, actually, here's the bottom portion of it, which I'm going to highlight right here, which is the same as, oh yeah, that portion, that's going to pick up protons, which is going to cause this portion at the top to rotate. When that part at the top rotates, and I'm going to show you a little model of this, that part at the top, when it rotates, it takes an ADP and an inorganic phosphate molecule, ADP and PI. It takes those two, puts them together, and forms ATP. It causes this pressure to accumulate, basically, and forces them together, and it's almost like it takes a piece of coal and makes a diamond out of it. Okay, so proton concentration is what's going to drive that. Now, this was a lot to process. The important parts of it all were the important parts of all of this is the important, sorry, the important parts of all of this is the steps of beta oxidation, and make sure that you understand what's taking place in beta oxidation. All right, well, I hope this is helpful, and if you have any questions, please don't hesitate to ask me.